This EGF pathway, short for epidermal growth factor pathway, is used for cell survival and proliferation, which is just growth and division. It can both regulate and stimulate growth. Usually, these EGF receptors are located in the skin cell. Because skin cells constantly grow and die out, ultimately the EGF pathway helps the cells grow by increasing protein synthesis and enlarging its cytoskeleton. Another way to help with cell survival is that the EGF pathway helps produce pro and anti-apoptosis proteins. More importantly, in cancer cells, EGF pathways are often dysfunctional. Therefore, many therapies target the EGF to treat cancer.
This pathway starts with the binding of the EGF ligand, NRG1 and NRG2, to the EGF receptor. Upon the binding of the ligand, the dimerization of the two EGFR parts happens where the two receptors join together. This allows for the autophosphorylation of the tyrosine kinases residues, where kinases cross-phosphorylate each other using ATP.
After the receptor is activated, the complex GRB2 and SOS will bind to the receptor. After the binding of the GRB2 and SOS, the RAS GDP will bind to the complex. This activates RAS by expelling a GDP and binding a GTP. This will lead to the activation of the RAF enzyme, which will then go on to activate protein kinases called MEKs.
They will then go on to the further activation of the regulated kinases 1 and 2, which is ERK1 and ERK2. The activated kinases eventually move into the nucleus to phosphorylate specific transcription factors like ELK1 and c-MYC, stimulating gene expression. This increases the rate of protein synthesis, which helps with the enlargement of the cytoskeleton, which leads to cell growth. Dimerization of the EGF receptor happens when the ligand binds to the active site and the two subunits join together.